forth an angel instead of a fiend. The drug had no discriminating action. It was neither diabolical nor defined. It but shook.
Guys, I'll be right back. 1 second. Louie, what are you doing, buddy? Hey, Louie. If I leave you out there, are you going to get in the litter box? Or stay there? Let's wait like another minute and see if we get a few more people. Let's just see. But any questions, concerns?
How long is gonna be the final exam?
What?
How long is gonna be the final exam?
It's about a third to double of what you've had. So it's about double what the regular lengths of the other exams, because it's worth about double the amount of points.
Okay, thank you.
Other questions? Let's jump into tonight, and we'll see if as we go, if we get more people. I'm sure we will. So. Like I said Monday night, if you were willing, if you were able to wait to jump into those lab exercises.
To remove their feelings and ego.
I have all of the figures that you're going to have edited and included in this PowerPoint. So all of the questions that have figures asking you to label or diagram or identify what's what, I have the answers here. If you'd be willing to turn on your cameras, I'd appreciate it. Thank you. Let's jump into tonight. So lab exercises 12 and 13. Bear with me. 12 and 13. Stop. 12 and 13. Muscular structure and function. So yeah, skeletal muscle structure and function. If you remember from the other night, skeletal muscle is striated with multiple nuclei. And the nuclei tend to be off to the side. And the striations are based on the fact that there are thin and thick myofilaments. What other type of muscle was striated? Do you remember?
Cardiac.
Right. So this is a longitudinal section. Where the dark lines are going to be the myosin. The thin line, the lighter lines are the actin by themselves, by itself. This is looking at it from a cross section, so not a longitudinal, but a cross section. So here's our muscle right here. That first slide that we were looking at was looking at it through here. Louis, I'm sorry, give me 1 second here. Louis, hey, come here. Here, Louis, here. Come here. Louis, hey, come here. Louis. Silly dog, come here. Louis, come here. Hey, come here. Go to tree, come on. Louis, come on, hey. Louis, now. Louis, here. Louis. Hey. I locked you
What
out.
did I say? Don't lock me
I
out.
locked with that one. That's why he barked.
Mary.
I'm in
Yeah.
class.
Ah.
I'm in class, Mom.
Okay.
Louis, come here. Hey, Louis. Louis, come here, silly dog. Louis, hey.
Okay, here.
Here, Louis. Oh, I tried that earlier and it didn't do anything. Sit. No, sit. Good boy. No, I'm busy locking my mother out, and that's why he's barking at the back door. Okay, I apologize. So that first slide that we were looking at was a longitudinal section going right down the center of the muscle. The second slide that we're looking at is a cross section. So like we talked about the other day, we're looking at bundles of fibers. So first one, looking at a cross section through the fibers, or a longitudinal section down the length of the fibers. These are looking at a horizontal section through the bundle. Showing the fascicles. Remember that we're looking at essentially a bundle of fascicles. And each of the fascicles is a bundle of myofilaments. Actually, let me say that differently. Each fascicle is a bundle of myofibrils. Inside each myofibril is a bundle of myofilaments. Endomysium wraps around each fascicle. Perimysium is going to wrap around the entire muscle fiber. So endomysium wraps around each fascicle. Fascicle. Each fascicle or fascicle is a bundle of myofibrils. Each myofibril is a bundle of myofilaments. So around each, and of course I know I messed it up. It didn't sound right to me. But that doesn't make sense. So a fascicle around each myofibril mycelium, rather than wrapping around. I'm sorry, I'm going to clear what I just did here because I knew I knew it sounded wrong when it came out. Endomycelium wraps around each myofibril. Each myofibril is a bundle of myofilaments. Each seal is a bundle of myofibrils. And the perimycelium wraps around each facile. And the epimycelium, just while we're on this topic, the epimycelium wraps around bundles of fascicles and can be continuous with the tendon. May be continuous with the outer tendon that is attaching the muscles to the bones. So endomycelium wrapping around a bundle of myofibrils. Perimycelium wraps around a fascicle, which is a bundle of myofibrils. Any questions? That wasn't very clear, so I hope that got better. So these are two figures that are coming from the lab manual, showing the same thing in a slightly different depiction. Perimycelium wrapping around the bundle of fascicles. The epimycelium wrapping around the outer edge and can be continuous with the outer tendon. endomysium wrapping around each muscle fiber, which is a bundle of myofibrils. I don't know, Mom. I thought she might be in your room because I can hear her barking. Maybe not. Any questions so far? Another picture coming specifically from the manual, showing the facile and how it's a bundle of muscle fibers, which are bundles of myofibrils. And each myofibril is a bundle of myofilaments, the thin and thick filaments. Around, and you can see it a little bit here, around the muscle fibers and in contact with the muscle fibers is the somatic motor neurons. Looking at the connection, the arrangement, remember that the functional unit of the muscle is a sarcomere. And one myofibril is essentially multiple sarcomeres attached end to end. So when we're looking at muscle contraction, no, you've got to let me work. These are my lazy Z discs, my really lazy drawing of Z discs. But each one of these, from one Z disc to the next Z disc, is the designation of one sarcomere. Next sarcomere, next sarcomere, next sarcomere, next sarcomere. Each sarcomere upon muscle contraction is going to shrink in length, meaning that this Z disc is going to come closer to this Z disc. When all of these are doing this, when all of the sarcomeres in one myofibril are doing this, overall the muscle is going to shorten. So the reason the muscle actually contracts and shortens in length is because each individual sarcomere within that muscle fiber is shortening in length, and they're attached to one another. So in some, the muscle contraction or the shortening of the muscle is a result of all of the individual sarcomeres that are attached end to end shortening at the same time. Is that clear? Maybe, since I can only see Justin, hopefully it is. So looking at a myofibril, it's one sarcomere attached end to end with other sarcomeres. Or actually, that wasn't a good way to say that. Sarcomeres attached end to end along the whole entire length of the myofibril. Within the myofibrils, we have the arrangement of the actin and the myosin, the thin myofilaments and the thick myofilaments. And they're attached to the Z discs. The way that we describe these, I'm going to try to do it on the whiteboard. We'll see what happens. Is I have these, I have a Z disc. And I'm trying to remember, because I am going through this same chapter with another class right now. I told you I teach physiology at Stevenson University. And guess what we're going over right now? We're going over muscle. And so I cannot remember what because we talked about it Monday night and I worked with them yesterday afternoon. I'm trying to remember what we covered in lecture and what subjects we haven't. And so if I go over something that you haven't heard anything about yet, please feel free to stop me. But so right here is one Z disc. One Z disc. Here is another Z disc. And in between these Z discs, we have an overlapping arrangement of thin filaments and thick filaments. I'm going to make our thin filaments, actually that wasn't what I meant to do, but it will undo, we'll make it undo. Okay, all right. I didn't mean to make that red, but we'll leave it red. We have thin filaments. Actually, let's do it a different way. Go out like this. And come out like this. Okay. Those are our thin filaments, and they're actually double coiled. So they're going to look like this. Do me a favor if you can, draw this out the same way I'm drawing it out. Hopefully draw it neater because you're not going to be trying to use your mouse to draw. But draw it out, because if you draw it out, you will remember it. And that's the reason I'm drawing it out here. So they're double coiled. Looks like a double coil of a spiral notebook. Those are my thin filaments. Which are actin. Here, Pacers. Come here. No, you got some. Oh, I didn't mean to hurt you. Come here. I'm sorry. Okay. And then in the center, I have thick filaments. I'm just trying to color that out. That didn't work very well. I'm trying to color this out. This little part right here, that was a mistake, but if I erase it, it's going to erase everything I just drew there. So this is my thick filament. And it's myosin. One thing that we'll also talk about is that this attachment point where the myosin attaches to the Z disc is called titin, because it was named titin because of its strength. So it is what attaches the thick filaments to the Z disc. Now, right here, from right here to right here, where we have just the thick filament, this is called the H zone. And from right here to right here, which covers the whole length of the thick filament right here, this is the A band. Do me a favor, before next week, memorize this structure, because we're going to talk about how each of these demarcations, like the H zone, the A band, and the next one that we're going to talk about, the I band, how they change upon muscle contraction. So one more thing that we need to talk about is from right here. Here's my next sarcomere. Oh, and of course it was supposed to be blue. It wasn't supposed to be that. Or actually, And then I have my thick filaments. That are attached to titin. And they run right along the length here. This area from right here to right here that is just thin filaments is called the I band. So just thin filaments called the I band. There's a reason I'm drawing this out, because if I draw it out, I want you to draw it out. And if you draw it out, you will remember it. You're much more likely to remember it. So this is my I band. Over the next several slides, we'll see multiple pictures of this. But if you draw it out, you're going to remember it. So A band includes the entire thick filament, even when it overlaps with the thin filaments. The H zone is just the thick filaments. The I band, that's just the thin filaments, and the middle of the I band is the Z disc, is just the thin filaments. One more demarcation. Right down the center of the H zone is the M line. And if it helps you to remember M line, think of it as the middle line of the sarcomere.
Professor, I have a question about the diagram.
Go.
The box that you drew with the green, I guess it's supposed to be a thick filament to the left.
Yes. Yes.
Now, did you say that was attached to the titin?
Yes.
Okay, so that line from there should go down to actually the titin instead of where you have it at the thin filament.
Where
On the left-hand side of the screen, right there, that the green line that you have attached to that there, right there. Should that go down to the titin or stop where you have it at the filament?
You know what? It depends on what figure you look at.
Okay.
As I just had students correct me because their figure showed that it was not like that in their book. It just recognize that what it's doing is it's connecting the thick filaments to the Z disc.
To the Z disc. Okay.
Yeah, good question. Because depending on what book you look at and what figure you look at, this might be, if you notice, I have the thin filaments on alternating lines. In other figures, it shows that they're on the same lines and they don't alternate. That's why I'm saying it depends on what you look at. Just recognize that the titin is attaching the thick filaments to the Z disc. No, I wrote it so that it wasn't alternating the other day in my class, and they were like, Professor Gessler, that's not right. That isn't right. And I'm like, why is it not right? And they showed me how the picture was showing that it wasn't alternating. I'm like, well, how do you know that's correct? It's a picture. But you know, we won't even bother.
Okay.
We'll bother arguing about what the figure says it is.
Okay.
So let's look, we'll see how it looks on the next several pictures because I have several different pictures showing the same structure. And then we can decide whether it's alternating or it should be on the same lines. Because according to this right here, actually, that's showing it's alternating too, maybe. Because, for example, looking at this, let me shift over. You don't see all of it here, but here's your Z disk. Here's your Z disk. Here's your titan that's connecting with a thick filament. This area right here is the H zone. That's only got the thick filament. The A band is all of this. The I band is just the thin filament with the titan. But no, nothing, none of the thick filament. Actin, like this picture depicts right here, is a double coiling of a globular of a strand. It looks like a pearl, a string of pearls, string of beads, doubled around one another, coiled around one another, with an additional coiling of tropomyosin, which is attached to troponin. The major reason I'm putting this whole figure up here is because it came right out of your manual. We're going to go through this piece by piece. But right now, understand that's the reason I'm putting it here is because it's one of the things that you're supposed to label in the manual in your lab exercises. And that's the reason I'm putting it here. Now, let me zoom out again. So specifically looking at the filaments of the myofibril. Myofilaments include myosin and actin. The myofibrils are bundles of these myofilaments that fill up most of the sarcoplasm. Remember, the sarcoplasm is the cytoplasm of the muscle fiber. Did we talk about this the other day? Justin's saying yes, but his mother is probably saying no. So we'll go. We'll say, did we talk about what sarco meant? Okay, we did. Okay.
Yes.
Sarco means
I have written down that it's muscle or flesh.
yes. Yes, that's what it is. So sarcoplasm is a cytoplasm within the muscle fiber. The sarcoplasmic reticulum is a specialized smooth endoplasmic reticulum that works as a calcium storage. And we'll see this in writing on another slide. And mitochondria are also present here because guess what? For muscle contraction, you need a lot of ATP. And so we have a lot of mitochondria around. The striated appearance is due to this ordered overlap of thin and thick myofilaments. Another word for the H zone is the bare zone, because it's just thick filaments. Let me add that to our drawing here. It's also called the bare zone. You guys, I think my one cat is locked in the bedroom, and I don't know if you can hear him, but I can hear him. And it will start to make me crazy. He's pining to get out of the bedroom. So give me 1 second to go and get him out. Hey, come on. Come on. Come on. You want to come out? Come out. Come on, you silly guy. Come on. Come on. Hey, come on. You're just being silly. Come on. Come on. Yeah, see, you're all right. Hey, I'm sorry. I don't know how well you could hear him, but it starts to make me crazy, especially when I know he really wants out. So bare zone, H zone is also called the bare zone because it's just the thick filaments. Yeah, you're doing all right. Did you hear him there? He is my little boy who was actually abused, and he was grossly neglected before I got him. And I've had him for about, how long has it been? 4 years. And he's come an exceptionally long way. And he's actually probably one of the most affectionate cat I have. He's a pretty cute little boy. And if you like Russian blues, meaning they're solid gray, and they tend to be hypoallergenic when they're purebreds, that's what he is. And so he's a stunning little boy. And if you can hear that high-pitched meow, he is probably part Siamese with that. We don't know. It's a lot of things, but he's a cute little boy. So striated appearance is due to that orderly overlap of the thick and thin myofilaments. The thick filaments are made of myosin, where the individual myosin molecules look like golf clubs. You have a tail made of 2 heavy chains and a head made of 2 light chains that are wrapped around one another. And the thick filament is a helical bundle of several hundreds of myosin molecules with the heads pointing outwards. The myosin is going to function as a motor protein. If you remember, we talked about motor proteins when we talked about retrograde axonal transport and anterior grade axonal transport. Myosin functions as a motor protein also, converting ATP to allow for motion. If you want to think of it this way, and we'll talk about it, we'll elaborate on it, but myosin is working like if you think someone playing tug of war, but doing it reverse. So tug of war, normally, how do you play tug of war? We've got people on 2 ends of a rope, and one group is pulling, one group is pulling, and we're all trying to push towards the other side, right? Or make them come to wherever we are, right? Am I right? Does this make sense? Okay. What we're doing here instead is the myosin is pulling on the actin and pulling the actin towards the center on both sides. So it's kind of an inverted tug of war. Rather than trying to pull things towards the ends, you're pulling them towards the center. That's the basis of the whole muscle contraction. Is that clear what I'm saying? So you're pulling on the thin filaments. Myosin is pulling on the thin filaments, moving it towards the M line. The thin filaments are going to be moving towards the M line as the myosin pulls on it. And the result is that because the thin filaments are attached to the Z disc, and the thin filaments are moving towards the center and getting pulled towards the center, the distance between the Z discs is shortening. So the whole sarcomere is shortening. This is kind of clear, maybe. Okay, it'll become clearer as we go, if it's not. So myosin functions as this motor protein, converting ATP into motion. The thin myofilaments are actin, where we have two intertwined strands. There are strings of globular actin subunits. And each actin subunit has an active site that can bind to the head of the myosin molecule. In relaxed muscle, this site is blocked by tropomyosin that has troponin attached to it. So underneath this little spot right here, this rope of tropomyosin, which is wrapping around these globular strands of actin, are spots where myosin binds to the actin. When troponin binds to calcium, the tropomyosin is going to shift away from those active sites and expose them, allowing us to allow myosin to bind to the actin. So our regulatory proteins here that are actually going to regulate whether or not myosin can ever bind to actin are troponin and tropomyosin that function as switches determining whether a muscle fiber can or cannot contract. Based on whether or not they are willing to expose the active site that myosin uses to bind to the actin. When calcium is present, troponin will bind to the calcium and it will shift the tropomyosin that's bound to the troponin. So contraction is activated by the release of the calcium from the sarcoplasmic reticulum into the sarcoplasm. Remember how sarcoplasmic reticulum is the storage for calcium. And it binds to troponin, and then troponin, when it binds calcium, changes shape and nudges the tropomyosin off the active sites that bind to myosin that are present on actin. So, and you know what, I think my students were right. I was doing it wrong the other day. I'll have to tell them that, because if this figure is showing the same thing that the other figures are showing, it's alternating. So here, the only thing that this figure is missing that we didn't draw, that we drew in on the whiteboard, is the Titan and the H zone, which is right here. So the myosin, when calcium is present, is going to bind to the actin, because the active sites on the actin will be exposed, and pull on the actin, moving both of these actin strands towards the center, the M line. And the result is that you get partial overlap of the actin at the M line when the muscle is fully contracted. What bands disappear when the muscle's contracted? What bands become smaller? Does the A band become smaller when the muscle contracts? It does. Is it, this is when it's contract, this is when it's relaxed. Does it
Yeah.
become smaller?
The I bands.
The A bands.
No, I'm saying the I bands become
I
smaller, right?
The I bands, you're right, the I bands do become smaller. Do the A bands?
No.
No, they don't. Does the H zone, is it still present when the muscle's contracted? Remember what the H zone was? It was this area right here. Is it present when the muscle's contracted?
No, because they're overlapping.
Right. So these are questions that are possibly good exam questions, because this is asking you to assess how well do you understand the sliding filament theory, which is what this is. That's why I said draw this out, because it'll help you significantly. Any questions about this figure right here? These are two pictures that are coming from the manual that are transmission electron micrographs of skeletal muscle, showing the I bands, the H zone, and the M line and the A bands. And that one sarcomere extends from one Z disc to the next Z disc. So each sarcomere is flanked by half of an I band on both sides and one full A band in the center and an M line.
Quick question.
Go.
I don't know if I missed this earlier, but do these like MZHIA, do they stand for something?
No,
Like,
not that I know of. They
okay,
just got named that.
I see.
If they did, I was not involved. I was not invited to
The naming party.
Yeah, I wasn't invited. You're right. I wasn't invited to the naming party when they came up with this. It's
Oh,
kind of, there's a lot of things where I wish I was involved in the naming party.
yeah,
But
it's weird.
let me give you an idea about how to remember with that idea. Thank you for saying something, Max. How are you going to remember that the I band is just the thin filaments?
There's two of them on either side, just like eyes.
Oh, well, that works. Oh, I hadn't thought about that one. That's a good one. All right. The other one is I haven't, let's come back and look at this one more time. Bear with me here. The eye bands here, and you can't see it. If we zoom in just a little bit more, do you see the little line that runs down the eye bands? Do you see that? Let me come in just a little bit more. You see the little lines that run down right here? The I bands are the lighter bands. The A bands are the dark bands. Now, the way your idea with regards to the eyes, which I'll remember that one because that's a good way to remember it, that you have two eyes and they flank each Z disc, just like you have two eyes that flank your nose. Another way to remember this is that the I bands, the word light, because these are the lighter bands in the striation, have an I in the word light. The A bands are the dark bands, and the word dark has an A in it also. So the I bands are light, the A bands are dark.
What made me think to ask was because the A bands are also like, that's actin, right? That
No,
makes
they're
those
not.
up?
It's myosin.
Oh,
Yeah.
okay. I see.
You're right. That would be a great idea if it was that way. I think it probably has some meaning. I don't know what it is. I'm guessing it means something. I can't imagine they would just come up randomly, give the names of the letter alphabet and not have it be named something. Give me one. You know what? Give me 1 second. Max, you like to ask me these questions that I don't know how to answer. But let me see. Just a sec.
Sorry.
No, it's all right.
Well, going along with what you said, the M line does make sense for, the middle or
Right.
medial. That's the only one that is obvious if it were to have a meaning.
I want to just see. I'm just gonna look really quick. Oh, okay. So if you look in your book, A stands for I knew there was a meaning once I thought about it. A stands for anisotropic. And I stands for isotropic, which refers to how these different bands reflect light. I'm not sure just how much that helps me, but that's where the names come from. I still think it's easier to just remember that the A is the darker band and the I is the lighter band, because the A is in the word dark and the I is in the word light.
Yeah, I'll stick with that.
Okay. So moving forward a little bit here. So the tropomyosin and the troponin are working like switches when they bind to the calcium. And when the troponin binds to the calcium, it exposes, it nudges the tropomyosin so that the tropomyosin reveals the active sites on the actin where myosin binds. And then myosin can bind and pull the actin towards the M line in the center when ATP is also present. Showing these, just the depiction of the I bands and the A bands and the H zone and the M lines and the Z discs. The titin is this huge, strong, springy protein that runs through the core of the thick filament, anchoring the thick filament to the Z disc and the M line. Titin because it's exceptionally strong. My niece has a pit bull named Titan because they're such strong dogs. So that's where the name comes from. So moving forward a little bit more, actually, yeah, let's move forward a little bit more. I have 2 videos, but let's move forward just a little bit more. Yeah. Actually, no, you know what, let's watch the first video now that I'm trying to remember what was in the, even though I only posted them a couple hours ago, I want to make sure that I'm giving you the right information. So this first one is just talking about this sliding filament theory or cross-bridge cycling. When the myosin binds to the actin, this is called cross-bridge cycling, having to do with the ATP breakdown. Trying to think, should I show this now? Let's wait, bear with me. I'm so sorry, I'm a little disorganized. But let me show you another. Let's continue with the PowerPoint just for a minute longer before I show you that. So muscle never contracts using that sliding filament theory. Muscle never contracts unless it's stimulated by a nerve. If nerve connections are broken, the muscle becomes paralyzed. So relaxation of the muscle is going to occur when the muscle impulses stop. Do you remember when we talked about synapses in the nervous system? What was a synapse? Synapse, S-Y-N.
Like how, signals basically are, sent through the nervous system and how the messages come across.
So here I have a neuron and I have an action potential that comes down. And there is a postsynaptic target. It can be another neuron, like in the case of the autonomic nervous system, or it can be a muscle or a gland. So when we're talking about nerve impulses coming down and exciting the muscle, they're doing it like this through synapses with the muscle. So here's the neuron. Presynaptic neuron. Here's the muscle. And we have that synaptic cleft just like we had with other synapses. So a motor unit is defined as the one nerve fiber and all of the muscle fibers that it synapses with and thereby innervates. So one motor unit is defined as all of the muscle fibers that are innervated by one nerve fiber. And it innervates them by synapsing with them. The muscle fibers of any given specific motor unit, so any of one motor unit, are dispersed throughout a muscle. They contract in unison, and they contract fully or they don't contract at all. That all or nothing, like we saw with action potentials. Muscle also has an ability to sustain long-term contractions as different motor units are taking turns contracting. They're alternating, even though different motor units are doing it, dispersed throughout the muscle. Some motor units are contracting while others are relaxing. We have small motor units, which are maybe 3 to 6 muscle fibers per neuron. So small motor units are defined as the fact that the neuron only innervates a few muscle fibers. Examples are your eye and hand muscles. We have larger motor units that require more strength than control. If you think about your eye and your hand muscles, we talked about writing and training your cerebellum. When you're using small motor units, you're working on your handwriting. Large motor units, you're running or you're lifting weights. Rather than trying to use fine control, you're using more strength. So you're producing powerful contractions that are generated by hundreds of fibers. For example, one motor unit, gastrocnemius muscle of the calf can have over 1,000 muscle fibers per neuron. So a synapse, like we said before, is this point where a nerve fiber meets its target cell. The neuromuscular junction is the synapse. So when we're talking about synapses in the case of muscle are, I don't know why I keep doing that. When we're talking about synapses, when we're talking about neurons synapsing with muscle, this synapse is actually the neuromuscular junction. Or NMJ. NMJ. So each terminal branch of a nerve fiber within the neuromuscular junction, so each axon terminal is going to form a separate synapse with each muscle fiber. And one neuron or nerve fiber can stimulate the muscle fiber at several points within the junction. If you look here, here's a synapse. Several different points. So the synaptic end bulb or the synaptic knob is the swollen end of the nerve fiber that contains synaptic vesicles of acetylcholine. You remember when we talked about what happens at a synapse, the release of the neurotransmitter? This is the same thing here, but rather than working from one neuron to another neuron, we're just looking at a neuron moving, connecting with a muscle. And the neurotransmitter that will always be used here is acetylcholine. So we have the synaptic end bulb, and then we have the synaptic cleft, which is the gap between the synaptic knob and the sarcolemma. Remember, the sarcolemma is the plasma membrane of the muscle fiber. The motor end plates is the area of the sarcolemma that's opposite the synaptic knob. Which contains acetylcholine receptors that when the acetylcholine is released into the synaptic cleft, it's going to diffuse across the cleft and bind to the acetylcholine receptors. The Schwann cells, what were Schwann cells? It's been a while, but what were Schwann cells?
They wrap around unmyelinated fibers in the PNS.
Right. And so in addition to their role in myelination, they're going to envelope and isolate the neuromuscular junction. So they're going to wrap around the neuromuscular junction and envelope it. So how does this work? It works similarly to what we saw before with at any synapse, any chemical synapse. The acetylcholine, so an action potential comes down. Once it comes down here, it opens voltage-gated calcium channels. Calcium diffuses into the synaptic terminal. And the acetylcholine is then released into the synaptic cleft. The acetylcholine then diffuses across the cleft and binds to the acetylcholine receptor. Well, we don't want to keep acetylcholine around for forever. And so it's going to get broken down by what's called acetylcholine esterase. But before it gets broken down, when it binds to its receptor, it's going to initiate a muscle action potential. So just like we saw action potentials in neurons, we see them in muscle too. They're probably not as complicated because they follow the same pattern. You have depolarization followed by repolarization. But the resting membrane potential is more negative. It's about minus 90 millivolts rather than minus 70 like it is in neurons. Because there are far more anions, negatively charged ions, on the inside of the membrane than on the outside. And just like we saw in the neurons, there's an excess of sodium in the extracellular fluid and an excess of potassium in the intracellular fluid. And the T-tubular membranes, did we talk about T-tubules the other day? Maybe. The T-tubules are indentations in the plasma membrane. Let's see, I'm right. Let's do it. All right. Yeah, right. Yeah, I'm sorry. I'm just trying to find the best picture of it. And this is a little misleading. I don't really like these pictures because they're not exactly showing you what you need to be able to see. But here's the T-tubules right here. Here's the myofilaments. We have that arrangement of thin and thick filaments. This T-tubule is an indentation that actually moves in to the muscle fiber. Moves into it. So it's an indentation of the sarcolemma that moves into the muscle fiber. So it's an indentation of the plasma membrane. So the T-tubule membranes contain more, this is the reason that the membranes are more negatively, have a more resting negative potential than neurons do, is because T-tubule membranes contain more chloride channels, which makes the cell more permeable to chloride, along with potassium. And the resting membrane potential is going to be maintained by the sodium potassium pump. Remember, that's the way we're always recharging the fact that we have more sodium outside and more potassium inside, is via that sodium potassium pump. Is this clear? So two things that are going to occur in muscle contraction. We're going to have an excitation, the process in which the nerve action potentials are going to lead to muscle action potentials. And excitation contraction coupling, where the events linking are going to, events that are going to occur that are going to link the action potentials that were initiated to activation of the myofilaments, preparing them to contract. So excitation is the action potentials along the muscle membrane from the neuron. Excitation contraction is that excitation of the action potentials being linked to contraction of the myofilaments, and they're sliding to allow the muscle to contract. So contraction, actually, okay, let's keep going for a moment. Contraction is the step in which the myofilaments are going to develop tension and begin to shorten. So we've got we've got 4 phases going on here. We've got excitation, where the nerve action potentials are going to result in muscle action potentials along the sarcolemma. And then we have excitation contraction coupling, where these action potentials along the sarcolemma are going to activate the myofilaments, preparing them to contract. Then we have a contraction, which is the step in which the muscle fiber starts to shorten. It develops tension and it may start to shorten. When the stimulation of the neuron ends, the muscle fiber then relaxes and returns to its resting length. So we have excitation, where the neuron is there we go. For four general steps of muscle contraction, or this whole process that we're talking about, where the neuromuscular junction is going to lead to contraction of the muscle. First one is excitation. Action potential coming down the neuron, or the let's say this, yeah, action potential in the axon, in the neuron, is going to result in an action potential along the sarcolemma of the muscle. That's our first step. The second one is excitation contraction coupling. Where the action potential that's moving along the sarcolemma is going to prepare the myofilaments for contraction. Preparing them to allow them to slide against one another and perform contraction of the muscle. What do you think we need? Do you remember what the switch was to allow for contraction to occur or not to occur? What was needed here? This excitation contraction coupling is what's going to result in the calcium release from the sarcoplasmic reticulum. Remember how that was the switch for whether myosin could bind to the actin release from SR or sarcoplasmic reticulum. 3 is a contraction, where the myofilaments are sliding against one another. In the sliding filament theory, sliding against one another, each other, and sarcomeres are shortening. Or is the relaxation. Calcium. Okay, so that the muscle relaxes, myofilaments relax. They go back to their relaxed length. Calcium is going to get pumped back into the sarcoplasmic reticulum. To the SR, allowing the muscle to relax. Those are the 4 steps. Sherry, I can tell you're writing those down. Let me know when you're done. Are you
Got them.
done?
Thank you.
Okay, awesome. So bear with me for just a few more minutes and then we will take a break. So excitation, the action potential comes down the neuron at the axon, and it opens voltage gated calcium channels in the synaptic node. Calcium enters and stimulates the release of acetylcholine from synaptic vesicles in the synaptic cleft. The acetylcholine then diffuses across the cleft. acetylcholine will bind to each receptor and open the channel, allowing sodium to come in and depolarize the membrane. Just like we saw with before with neuronal action potentials. Sodium is going to enter, shifting the resting membrane potential from -90 to +75. Potassium is going to exit and return the membrane potential back to -90, repolarizing it. So this is depolarization, the first sodium entering. Potassium moving back out is repolarization, just like we've seen before. This quick voltage shift is called, rather than calling it a action potential along the sarcolemma, to distinguish it from action potentials that occur in the neurons, we call this an end plate potential. Because remember, this is the motor end plate where this is occurring. This is a motor end plate where it's occurring. So we call it an end plate potential that's being initiated here. Voltage changes in the end plate region are going to open nearby gated channels, producing the muscle action potential that spreads over the sarcolemma and travels along the T-tubules. Remember how those T-tubules are indentations of the sarcolemma. The action potentials are going to spread down the T-tubules, opening voltage-gated ion channels in the T-tubules and calcium channels in the sarcoplasmic reticulum. So calcium leaves the sarcoplasmic reticulum and enters the sarcoplasm. It's going to bind to troponin on the thin actin filaments. Calcium is going to bind to the troponin. And remember what happens when that happens. The troponin shifts in its configuration and pulls the tropomyosin off the active sites on actin that myosin binds to. Then we get
I
this.
have a question.
Go for it.
I'm sorry.
No,
So
go.
when somebody has a heart problem and they're doing like troponin check,
They're
how
doing
do they
what?
do that then?
They're doing what?
Sorry, hold on. Let me put my volume up. So when somebody has like heart problem, like, you know, when they're complaining of chest pain or any heart related issue and they're doing like troponin check, how do they do that? Like, you know, when they
Troponin
check the
check.
labs, I'm sorry, what was that?
A troponin check.
Yeah. They're checking like their troponin level, but how do they check that then? Like, I guess just like they would with like calcium and potassium, they check it like that, but they check specifically for troponin level.
You asked me this and I don't know because I don't know. But let's do troponin checkup. A troponin test measures the levels of troponin T or troponin proteins in the blood. These proteins are released when the heart muscle has been damaged, as occurs with the heart attack. When the muscle gets broken down, it's going to release the troponin out. the more damage there is to the heart, the greater the amount of troponin T and I that will be in the blood. Louis, really? Come here. Louis, shh. Hey, Louis. Ah. Louis. You silly dog. Did you hear me kind of almost say something colorful? Louis, come here. Troponin, get over here. Louis, come here.
Did you lock your mom out again?
I hope not. You heard that too. I was like, mother, yes, getting me in trouble. I'm really good. You know, I lived alone for a long time and I'm really good at locking doors. And she's like, Mary, don't lock me out. And normally I'm pretty good about not doing it, but not tonight. All right. So that's what they're looking for, is they're using it as a measure of how much heart damage there is to the muscle. It's a good question because I've never thought about it. Does that answer the question, Zena?
Yeah, I was just wondering like what, like how they do it. Like I was just like I'm sorry.
I don't know.
It's okay. Maybe I could like YouTube it or
I'm
something.
wondering if they use an antibody that binds to troponin. And when they do that, they can measure how much of the antibody troponin complex settles out. That would be my guess.
Okay.
This is a question for my mother, but that would be my guess because that's the way I would do it.
Gotcha. Thank you.
The way Alan and Michael said they would do it too, like it would have to like almost settle out because it's not, that's what they're not looking for like a protein, or it's like, it's what would settle out in the blood.
How do they get it to settle out? Do they do it with an antibody?
Not a clue.
Okay.
Wait, what was the question?
When they're looking at troponin levels in the heart, how do they measure it?
So I believe they do a lab draw, and the lab uses a from what I have a friend that works in the clinical lab setting. She works for my former hospital, and she said that they had a specific substance that they used to put the blood work on the slide. I don't know the technical term, but basically they would put it on the slide and they would look to see the troponin levels. But they got a new machine out there now that automatically does it. Like they put the blood there and it reads it automatically.
Yeah, Michael just corrected me because it is a detectable protein. It's the lab equipment is able to just read it automatically. And if you have the elevated levels in your blood, your patient has probably had either a myocardial infarction or something that has caused some major heart damage, because that's the only way it's going to release into the bloodstream in those levels.
And it shows up on I believe not the troponin levels, but like the MIs and everything, they show up on like an EKG. So I know typically
Myocardial infarction.
So I know at UPenn, I just started there. I know our policy is if a patient has had a cardiac event of some kind, they automatically draw the labs to see what the troponin levels are and to see where like all the levels are, to see what's going on, to see what caused the event.
Okay. You know what? Because I want to know what they're using to detect it. I mean, I can understand that they have a reader, but I would think that they need some way antibodies. So specific antibodies against troponin have enabled the development of assays that can measure the concentration of troponin in the blood. So yes, they're still using antibodies because that's just an easy way. We use things that are specific to binding to proteins. And that allows it to settle out. When you go into 122, if you had me, even though Zena will tell you we're not going to finish all the chapters in 122. If you had me, we go over blood first. And I like to go over blood first because I think it's probably one of the most important. And when antibodies bind to proteins, that will pull them out of solution. And that will pull them out of the blood. So Zena, just be reassured that even though we're not going to finish every chapter we were supposed to go over in 122 because I'm slow, we're going over the more important chapters according to Professor Gessel. So
Thank
yes,
you all for answering that question for me.
you're welcome.
That helped
It was a good question. I was like, I don't know why they measured troponin levels. Let's see if I can get what I had on this. Okay, so cross-bridge cycling involves the hydrolysis of ATP. Remember that the myosin heads are going to hydrolyze ATP. Going to hydrolyze ATP and get energized when they hydrolyze ATP. They hydrolyze ATP, and this puts the head into a configuration where it can then bind to the actin. It binds to the actin, and then it releases the ADP, the adenosine diphosphate, after the hydrolysis of ATP. The release of the ADP makes the head swivel back. Pushing the actin forward in one direction. So it pushes the actin forward until it binds another ATP. The myosin is stuck to the actin in that pushed forward state. That's the basis of rigor mortis. And we'll talk more about that next Tuesday. But so ATP hydrolyzes, the myosin hydrolyzes the ATP, and it puts it in a high energy state where it binds to the actin. It then releases the hydrolyzed ADP and phosphate, and that release cocks the head of the myosin, pushing the actin forward towards that M line, which would be like right here. When the myosin binds the next ATP, it can detach from the actin. It'll then hydrolyze the ATP again and start over again. So the myosin is going to pull on the actin, causing the filaments to slide forward towards that M line. As a result, the Z discs move forward toward each other and pull on neighboring sarcomeres, resulting in an overall shortening of the myofibril. Remember, as individual Z discs are moving towards each other, we're shortening one sarcomere at a time. And because they're attached end to end, the overall process is that the whole myofibril is going to shorten in length. So thanks to structural proteins, there's a transmission of force throughout the entire muscle, resulting in the whole muscle contracting. So the individual lengths of the thickened filaments are not changing. It's the overlap that's causing the change in the length. Bear with me as we let me keep going for a minute longer, and then we'll take a break, I promise. Relaxation, so nerve stimulation stops. Acetylcholine release stops if nerve stimulation stops. Acetylcholine esterase will break down whatever remaining acetylcholine there is, and the fragments will be reabsorbed. The calcium will be pumped back into the sarcoplasmic reticulum by the active transport. And the calcium in the sarcoplasm will bind to calsequestrin and be as it's stored in the sarcoplasmic reticulum. And once the calcium is gone, the tropomyosin moves over the actin's active sites, and myosin can't bind to actin anymore, allowing the muscle to relax. So calcium is pumped back, tropomyosin reblocks the active sites, and the muscle fiber ceases to produce or maintain tension. And the muscle fiber returns to its resting length. Now, one more thing, and then we will take a break, I promise. Let's watch 2 quick videos describing this maybe a little better. I wanted to finish that before we took a break. So bear with me. Let's watch this first one, and then we'll watch the second one.
I have to stop and tell you guys.
So this is talking about excitation, contraction, coupling.
Skeletal muscle contracts only when stimulated by nerve impulses from a motor neuron. Without innervation, muscles become paralyzed and eventually atrophied. The axon of a motor neuron usually gives out many branches, supplying multiple muscle fibers. These fibers contract in unison when activated and constitute a motor unit. A motor unit can contain anywhere from just a few muscle fibers to thousands of them. Small motor units are found in muscles that require finer control. For example, muscles that are responsible for subtle movements of the eyes. Large motor units are found in larger muscles that require strength, such as muscles of the arms and legs. Muscle fibers of the same motor unit are usually not clustered together, but scattered throughout the muscle. This way, stimulation of a motor unit has a slight but uniform effect on the whole muscle, and not causing a small, localized twitch. The strength of a muscle contraction is determined by the number of motor units that are activated at one time. Even at rest, most muscles are in a partial contraction state called muscle tonus, which is maintained by alternating activation of a small number of motor units. The connection between a motor neuron and a muscle fiber is called a neuromuscular junction, which is basically a chemical synapse between the nerve terminal and a specialized area of muscle cell membrane called the motor end plate. When an action potential reaches the nerve terminal, it causes the release of the neurotransmitter acetylcholine into the synaptic space. Acetylcholine then binds to nicotinic receptors on the end plate. Nicotinic receptors are ligand-gated ion channels. Upon binding to acetylcholine, they open to allow sodium to enter the cells, depolarizing the cell membrane, producing the so-called end plate potential. An action potential is generated in the muscle cell only when the end plate potential reaches the threshold required to activate voltage-gated sodium channels located outside the end plate in the neighboring membrane. When activated, these channels allow faster influx of sodium, further depolarizing and eventually reversing the polarity of the cell membrane. At this point, voltage-gated potassium channels open for potassium to move out, quickly returning membrane voltage to its original resting value. Once generated, the action potential spreads like a wave, thanks to similar voltage-gated ion channels located throughout the muscle fiber. The action potential also runs deep into the fiber via T-tubules to reach the sarcoplasmic reticulum. Here, it activates voltage-gated calcium channels, releasing calcium from the sarcoplasmic reticulum into the cytosol of muscle cells. Calcium then sets off muscle contraction by the sliding filament mechanism. This mechanism is described in another video. Another important component of the neuromuscular junction is the enzyme acetylcholinesterase. This enzyme removes all acetylcholine molecules that do not immediately bind with the receptor, and those that are done activating a receptor. The enzyme action essentially terminates synaptic activation, giving the muscle time to relax, and thus preventing continuous contraction that would result in muscle spasms. Substances that cause muscle weakness or paralysis do so by interfering with the function of neuromuscular junction. Botulinum toxin prevents acetylcholine release from the presynaptic side of the junction. Some other toxins attach to nicotinic receptors, blocking acetylcholine from binding but do not open the ion channel. Certain drugs lodge into the channel of nicotinic receptor, blocking the passage of sodium. All these substances prevent activation of muscle cells and cause flaccid paralysis. On the other hand, some pesticides inhibit acetylcholinesterase, preventing degradation of acetylcholine, causing continuous activation of muscles. That's how they induce muscle spasms and cause spastic paralysis.
All right. I think we'll end that there. Let's watch one more video. It'll be a faster one and then we'll take a break.
If you're in medical school, I can guarantee anatomy is going to be the hardest class to take.
Muscle contraction is at the basis of all skeletal movements. Skeletal muscles are composed of muscle fibers, which in turn are made of repetitive functional units called sarcomeres. Each sarcomere contains many parallel overlap.
Bear with me. I'm going to start at the beginning now that it's enlarged, because I want you to see this a
Muscle
little better.
contraction is at the basis of all skeletal movements. Skeletal muscles are composed of muscle fibers.
I'm sorry,
which
be
in
patient
turn
with
are
me.
made of repetitive functional
Dismiss.
units called sarcomeres.
Stop. I want you to be able to see it all, and that's why I'm doing this, so bear with me just for a sec. Okay. Come on. All right, now enlarge and push play.
Sarcomere contains many parallel overlapping thin, actin, and thick myosin filaments. The muscle contracts when these filaments slide past each other, resulting in a shortening of the sarcomere and thus the muscle. This is known as the sliding filament theory. Cross-bridge cycling forms the molecular basis for this sliding movement. Muscle contraction is initiated when muscle fibers are stimulated by a nerve impulse and calcium ions are released. The troponin units on the actin myofilaments are bound by calcium ions. The binding displaces tropomyosin along the myofilaments, which in turn exposes the myosin binding sites. At this stage, the head of each myosin unit is bound to an ADP and a phosphate molecule remaining from the previous muscular contraction. The myosin heads release these phosphates and bind to the actin myofilaments via the newly exposed myosin binding sites. The two myofilaments glide past one another, propelled by a head-first movement of the myosin units, powered by the chemical energy stored in their heads. As the units move, they release the ADP molecules bound to their heads. The gliding motion is halted when ATP molecules bind to the myosin heads, thus severing the bonds between myosin and actin. The ATP molecules are now decomposed into ADP and phosphate, with the energy released by this reaction stored in the myosin heads, ready to be used in the next cycle of movement. The myosin heads resume their starting positions along the actin myofilament and can now begin a new sequence of actin binding. The presence of further calcium ions will trigger a new cycle.
Oh, no, let's let him finish. Okay. Let's take 7 minutes here and come back. And so my clock says that it's 7:59, so let's come back at 8:06.
Hi everyone, Dr. Mike here. In this video, we're going to talk about everybody's favorite physiological concept when it comes to skeletal muscle, which is the sliding filament theory. But before we begin, we need to
My students asked if Louis started barking again because I locked you out again. No, they heard you say, you locked me out again. I was like, yep, so that was me getting told on. Huh, were you barking? Well, it looked like it was. I was like, you can get lost in that. Sure is pretty though. Sean said, well, just keep it so healthy. Did you laugh? Gone for 2 weeks and this happened. But I told her I didn't get to Friday. But if I have to go get a new license, Friday's today. I checked with police and see which side that. She said, no, she said something about work. And she was on that Friday morning, they were going to go to the temple. And she said she was going to be doing it. That's right. Delia's yard. So you did Delia's yard too? I can't resist. If I have any energy left at all. Besides, I want to cut down enough. It's such I don't want their yard. No, I have to go over, sneak over. So it's not completely altruistic. You're doing it. It's selfish. Yep. Anyways. But at least you admit to it.
must have something going with it. You don't like that. Oh, maybe it's down. Oh, yeah, there's quite a bit of trash. Couldn't do one more thing tonight. I know one day it was there on that day it wasn't. They did a pretty good job cleaning it up considering the heat it was. But I'll have to go out and do some of that. They're just little pieces of lines, you know, that over it off the front. At one point I knew I didn't know. I'm gonna go to class out here all wow. So long time out there. New. Or work out the yard without gloves. The knife will be on part, all my muscles scare the heck out of me. And all my students laugh. It's loud. You have quite a convincing argument. Yes, and I have to do it.
So he's excited.
Even though I came in and ate more and eating chocolate.
He's excited about you needing to do his hair.
And I have to make it without the color. Which is awful. All right.
Come here. Come here.
The food that I grow,
Yeah,
I'm well enjoying it.
good.
Tomorrow I'm going to go buy some ground round. A better buy than the past.
All right, let's actually wait another minute. Let's wait another minute. Okay, so overview from beginning to end. The nerve impulse arrives at the axon terminal of the motor neuron and triggers the release of acetylcholine. Acetylcholine is going to diffuse across the cleft, bind to the receptors in the motor end plate, and trigger a muscle action potential. Acetylcholine esterase will destroy the acetylcholine so that another action potential does not arise unless more acetylcholine is released from the motor neuron. As that end plate potential moves across the sarcolemma, it's going to move down into that indentation, the T-tubules, and open calcium release channels in the sarcoplasmic reticulum, which allows for calcium to flood the sarcoplasm. Calcium binds to the troponin on the thin filaments, exposing the binding sites on actin that myosin binds to. The result is that we get contraction, which includes power strokes of the myosin that use ATP. The myosin heads bind to the actin and swivel and release with the hydrolysis of ATP. The result is that the thin filaments are pulled towards the center of the sarcomere or the M line. When the muscle starts to relax, the calcium release channels in the sarcoplasmic reticulum close, and the calcium active transport pumps use ATP to restore the low level of calcium ions back to in the sarcoplasm. And the troponin, tropomyosin, then slides back into place, covering and blocking the myosin binding sites on the actin. And the muscle relaxes. Any questions about this? Mom? Mom, will you grab her jacket? Sorry, my little poodle here who got cut recently is here freezing in my lap. And it's making me feel bad, so we're going to grab her jacket. Makes me feel bad when they're cold. So the strength of muscle contraction depends on the size and number of the motor units. Remember that we have big motor units and we have smaller motor units. And remember that one motor unit is defined as all of the muscle fibers innervated by one motor neuron. So larger motor units are going to generate more tension because there's more muscle fibers contracting. As a greater number, my mom will do it. Okay. As a greater number of motor units are activated, the muscle can generate more tension and lift heavier loads. Remember what's regulating what motor units are being innervated and what ones are not. Do you remember? Which motor units are being activated and which ones are not? This is the cerebellum cross-talking with the cerebral cortex and the basal ganglia, saying this is, you go to pick up a heavy box and your cerebellum is going to determine what motor units get recruited and when, whether you use smaller ones or whether you use larger ones. So when the motor unit contracts or gets excited, all of the muscle fibers in one motor unit are going to contract in unison. And the muscle fibers of the muscle unit are dispersed throughout the muscle. They're not clustered together. So the strength of any muscle contraction depends on the size and the number of motor units involved. So motor unit recruitment is a process by which the number of active motor units increases. The weakest motor units are recruited first, followed by stronger motor units. Motor unit summation is when the recruitment of additional motor units generates more force or more contraction. As more motor units are activated with increased stimulus, you generate more contraction. So motor unit summation is essentially the strength of one motor unit, its contraction being added to the strength of the contraction of the other motor units. Just like when we are looking at summation to look at graded potentials, whether or not we'd initiate an action potential, we're doing the same thing here. Motor units contract alternately to maintain a contractions for longer periods of time. So just like when you go to move a car, you recruit more people to push heavier loads. And once enough people are recruited, no more people are needed. So you're going to recruit more and more motor units. The weakest ones get recruited first until you have enough motor units recruited to perform the action you need. So maximum tension depends on a couple different things. The rate at which the nerve impulses are arriving at the muscle, the size of the motor units, and one thing that we'll talk about next week, the amount of stretch before a contraction. How do skeletal muscles meet their energy demands during rest and exercise? This has to do with the nutrient and oxygen availability. The two things that are really needed here are glucose to make ATP and oxygen to make that ATP from glucose. So muscle contraction demands incredible amounts of ATP. Remember, each myosin head is requiring 1 ATP per swivel of the head. So a contracting muscle cell can use up to 2 million molecules of ATP every second. And without the constant supply, muscle contraction ceases. If you remember from that video, the myosin is stuck to the actin until another ATP binds. The reason you get rigor mortis, and we'll talk about this next week, but the reason you get rigor mortis is because ATP production stops and the myosin is stuck to the actin in a stiff, contracted position. The calcium leaks. It leaks out into the sarcoplasm because things start to disintegrate. And so now all of the myosin heads are exposed. All of the myosin, active sites on actin are exposed to myosin because we've got this major leakage of calcium, but we have no ATP. And so everything is stuck until the filaments start to disintegrate, resulting in the breakdown of rigor mortis. I know you wanted to know that, but you heard it anyway, and we'll talk about it again next week. I think it's interesting. So 2 main pathways of ATP synthesis, because we do need a lot of ATP. The first one is aerobic respiration, which is going to produce far more ATP and does not generate lactic acid, but it requires a continual supply of oxygen. Aerobic meaning oxygen required. after we start running low on oxygen, because it doesn't take very long, anaerobic fermentation will enable the cells to produce ATP in the absence of oxygen. This yields very little ATP, but generates lactic acid. So how do we get the oxygen that we need and the ATP that we need? Oxygen is briefly supplied by myoglobin. Remember, hemoglobin, which binds to oxygen, myoglobin is a derivative of hemoglobin that's found in muscle. Remember, this is what gives muscle its red pigment. It's what makes dark meat look dark. So muscles that tend to be more actively used have a lot more myoglobin in them because you need more oxygen. But it does get rapidly depleted. There's two enzyme systems that are going to control ATP production. A myokinase. Did we talk about what a kinase was? Let me do something really quick. A kinase, any kinase, any kinase. Any kinase adds phosphate groups to something. Remember that phosphate functional group that we talked about in like chapter 2, really, really long time ago? They add phosphate groups to lots of different things. In contrast, phosphatases remember that anytime you see that word ASE on the end of something, that means it's an enzyme. So phosphatases remove phosphate groups. So that's the difference between those. So a myokinase is going to work in muscle to add phosphate groups to ADP. Because that's what myo means, right? Muscle. So myokinase is going to transfer a phosphate group from one ADP to another one. Remember what does ADP mean?
Adenosine diphosphate.
And what does that mean?
It means 2 phosphates.
That's what it means, 2 phosphates. What's the difference between that one and adenosine triphosphate, Justin?
1 phosphate.
Triphosphate?
Yeah, that's 2. That's 3.
Oh, yeah, you're right. Okay, you're right. So if we remove, so myokinase is going to take 1 phosphate group from one ADP and add it to another ADP, making an ATP. That's one way that we can make more ATP once oxygen gets depleted. Another way that we can make ATP is creatinine kinase. Got a lot of kinases here. Creatinine kinase, which is going to transfer the phosphate group from an enzyme called creatinine phosphate and give it to another ADP to make another ATP. So we've got two different ways that we can produce more ATP outside of using anaerobic respiration when we've depleted all the oxygen that we have, or when we're getting really low on it. So muscles will meet ATP demands by borrowing phosphate groups from other molecules and transferring them to ADP. This combination of ATP and creatinine phosphate provides nearly all energy for short bursts of activity. You guys, if this isn't completely clear, we're going to be looking at this again on Tuesday. For short-term energy, so rather than a sprint of 8 to 10 seconds, for 1.3 to 1.6 minutes, short-term energy. As the muscle activity continues, the creatinine phosphate levels are going to get depleted. And the muscles obtain glucose from the blood and their own stored levels of glycogen. Remember, glycogen is a stored form of glucose. In the absence of oxygen, glycolysis can only generate 2 ATP for every glucose molecule consumed, and lactic acid is produced. For long-term energy, like 15 kilometers, after about 40 seconds, the respiratory and cardiovascular systems start to deliver oxygen fast enough for aerobic respiration to meet the demands. So your heart and your respiratory system have to get up to gear in order for you to use aerobic respiration solely. Your heart has to get pumping fast enough. Your respiratory system has to start, you have to start breathing faster in order to get to where they need to meet to allow for aerobic respiration in the muscles. So these two here, intense exercise and then short-term energy, is what we will do until the respiratory and the cardiovascular systems can supply enough oxygen to allow us to work with aerobic respiration and produce more ATP. Is this clear? All right, Zena, is this clear? Because you're none of you three that I can see are actually nodding your heads. Is it clear? Is it not clear? It's okay to tell me it's not. So we have to be able to, we don't have a lot of ATP. ATP is not stable. It doesn't stick around very long. And the myoglobin initially will give up all of its oxygen. But then it gets rapidly depleted. It stores oxygen, but it doesn't, but we start burning through it really, really fast. And so we need two systems that will start generating ATP long enough to maintain us until the cardiovascular system and the respiratory system are able to provide enough oxygen to the muscles. and allow us to start using aerobic respiration. So the first one is these enzyme systems that are going to be transferring ADPs, are transferring phosphate groups from one ADP to another ADP in order to produce ATP. Or they're taking the phosphate groups from creatinine phosphate and adding them to ADP to produce ATP. In the second one, the muscle will also use whatever glucose stores it has and whatever glucose it can obtain from the blood. But without oxygen, you can only produce 2 ATP from every glucose molecule. So we're still waiting. We just have to make it long enough that our cardio and our respiratory systems get up to where they need to be to supply the muscles with enough oxygen. Because once we move into aerobic respiration, because the cardiovascular and the respiratory system are able to provide enough oxygen, rather than only generating 2 ATP per glucose molecule, we're now going to produce 36 to 38. But we have to have the oxygen to do that. Is it a little clearer now? So for 30 minutes, about 30 minutes, energy comes from equally from glucose and fatty acids. After 30 minutes, the depletion of glucose causes fatty acids to become the significant fuel. We talk one of the deficits to AMP for HAC, and don't tell any of the other professors I say this at HAC. This is what I get for teaching at a couple different schools, is they move you right into AMP 1 and 2 without having any, many of you don't have any very little biology background before you show up in this course. And if we had more biology background, you would know what all this was right here before we showed up here. Because this is why I teach Biology 101 at Carroll. And though a lot of those students are going to go into AMP next. But what's amazing is how little of them actually remember this. And I think you know, it's easy for me to say that now that I've taught it for a couple of years, because it comes to me really easily. It doesn't come so easily to them, and I understand that. But more than anything, understand the ATP is required. And after 40 seconds, your respiratory and your cardiovascular system are going to produce enough ATP, provide enough oxygen that you can produce enough ATP that you're not transferring them from one molecule to another ADP and just depleting your sources of ATP. Give me a chance to see if I can't find a short video that would explain this a little better, because I don't like the way I'm explaining it here between now and next Monday. just for right now, because of your exercise questions, let's move forward for right now. Due to muscle fatigue, what is muscle fatigue? Couple different things that can cause muscle fatigue. An inadequate release of calcium from the sarcoplasmic reticulum. Well, you know, the sarcoplasmic reticulum doesn't have infinite stores of calcium. Eventually, it's going to slow down. Another one is depletion of that creatinine phosphate, oxygen and nutrients. So we're running out of glucose, we're running out of creatinine phosphate, and we're running out of oxygen. So muscles are going to fatigue. Buildup of lactic acid and ADP, because we can't make ATP if we can, even though we have a lot of ADP, unless we have things to transfer phosphate groups to it, we're not going to make more ATP. And what if we start having less acetylcholine around in the neuromuscular junction? That's going to cause muscle fatigue if we have less acetylcholine. Why do you continue to breathe heavily for a period after stopping exercise? This need to breathe heavily is what we call an excess post-exercise oxygen consumption, where you breathe heavily in order to meet a metabolic demand known as the oxygen debt, which is the difference between the elevated rate of oxygen consumption following exercise and the usual resting rate. Extra oxygen, this breathing heavily to bring in more oxygen, is going to work to restore your metabolic conditions back to the resting state. You need to build up those myoglobin oxygen stores again. You have to build up your creatinine phosphate stores again. The only way for you to do that is by getting more your oxygen levels back, so that you can get ready to do it all over again. So this excess post-exercise oxygen consumption is needed to aerobically replenish the ATP, help regenerate some of those creatinine phosphate stores. Zena, it looks like you have a busy household there. I don't know how you guys do that sometimes. Zena, I'm gonna break all FERPA violations. She has a 5 and a 6-year-old, and they're 10 months apart. I don't know how she does it, but all right. I hope I don't get in trouble. Probably get in trouble, maybe. To replay, so EPOC is going to replace oxygen reserves on the myoglobin. So replenish ATP, replenish creatinine phosphate stores, replenish the reserves on the myoglobin. And to deliver oxygen to the liver that's busy trying to get rid of the lactic acid that you've built up. And to provide oxygen to many cells that have elevated metabolic rates after exercise. This excess post-exercise oxygen consumption can be at least 6 times your basal amount of breathing, your normal amount of breathing, 6 times higher than your normal amount of breathing, and lasts for about an hour. And even though that was probably, especially this slide, was a lot more than you already knew before you came in this class, you will get through AMP2 and you'll be fine. Zena can tell you that she's taking 122 with a lot of students who had me for 121. And somehow they're doing just as well in 122 as they did in 121, which is just as well as you guys are all doing. And I think at the very end of your chapter, there, which Zena is not going to get because I'm running out of time in the semester. And this is the first time I've taught 122, as Zena can tell you. If you think your class is chaotic, try that one out. But at the very end, in the chapter on nutrition and metabolism, they go through all this. And so you will see it again. Right now, just recognize that we have to get aerobic respiration. We have these other phosphate systems that are going to allow us to produce ATP until our respiratory system and our cardiovascular system get to where they need to be to produce enough oxygen. That's the gist of it. And you guys, that is the end for tonight. Let me know if there's questions on this lab exercise that are harder than normal and that you're not sure how to answer, and I will help you with them next week. Okay.
Thanks. Have a good
Yeah.
night.
You too.
All right, thank you. Have a
Thank
great
you.
night.
Of course.
Have a good night.
Thank you so much.
Have a good night.
You too.
Have a good night.
You too.
Thank you.
Of course. Savannah, are you there?